to have Drs. Moorbeck and Bonneau from Senckenberg and the National Oceanography Center in Southampton report on the work that we've sponsored. So we've gone on 2 cruises so far. Thank you.
So good afternoon, and thank you, Ralph, for introducing us. This is a joint presentation together with ORV, and I will start the presentation, and I will give you a report on the status of uksrl's activities on metazoan and myofaunal baseline studies in the UK1 claim area. So let's start by looking at the sampling areas. On the left, you can see the UK1 claim area, and today we have focused our sampling on 2 strata, 2 30 by 30 kilometer study areas, because previous studies suggested a 30 by 30 kilometer stratum is large enough to include a wide range of environmental parameters, such as variable nodule deposits and topography, and small enough for intensive sampling during a 30 to 40 day cruise. Sampling was conducted during 2 cruises of 30 to 42 days at 10 to 12 random locations in each stratum. UK1 stratum A was sampled during Abyss Line 1 in October 2013 aboard the research vessel Melville, with 10 out of 11 successful megacore deployments. From each deployment, 3 to 4 cores were selected for quantitative statistical analysis, providing 39 cores in total, and 1 core was selected for molecular analysis, yielding 11 cores in total. UK1 stratum B was sampled during Abyss Line 2 in February-March this year aboard the research vessel Thomas G. Thompson, with 12 out of 13 successful megacore deployments. Here, 4 cores were selected for quantitative analysis, providing 48 cores in total, and 1 core was selected for molecular analysis, providing 12 cores in total. And Oselbauer's and Connelly megacore, an updated version of a multicore, was used to collect 12 10 diameter tube cores of nearly undisturbed sediments and topwater. And the first 5 centimeter sediments were fixed in 4% borax sulfate formaldehyde solution for quantitative analysis or DES for molecular analysis. The nodules present inside the cores were removed, rinsed over a sieve, and fixed separately from the sediment. So we used the same sampling procedure as my colleague Annika Janssen did for the German claim area. All nodules were photographed, measured, and tested gently. The myofauna was extracted from the samples by centrifugation using Levasil kaolin, and the separation into major taxa was done by hand sorting under a light-saline microscope. So this slide looks almost the same as the slide of my colleague Annika Janssen, but it is not the same pie chart. It looks really similar. But we have much lower abundances in the, as in the German claim. Here the total myofauna, when I speak for total myofauna, I mean total metazoan myofauna, but I will not say this every time, so because this talk is about metazoan myofauna. Okay, the total myofauna abundance ranged from 112 to 237 individuals per 10 square centimeter. And typically the Nematoda were the most dominant taxon, ranging between 82 and 89% of the total myofauna abundance. Second most abundant taxon were the copepods, accounting for 9 to 14%, followed by the annelids with less than 2%, and other taxa such as ostracods, luciferans, and eucaridans, which account together for less than 2% of the myofauna. Regarding the community position at the different stations, no significant differences were found in major taxa. We also made comparisons between the stations and myofaunal densities, and the highest mean abundances were reported at station H and station K, with about 235 individuals per 10 square centimeter, and a remarkably low mean abundance was found at station C, with approximately 91 individuals per 10 square centimeter. And the densities of station C, I would say, differ marginally significant to station H and K, with a p-value being just over the threshold for significance. These results correspond quite well with the sediment parameter analysis conducted by Carlos Neira from the Scripps Institution of Oceanography. He found that at station C, where we had the lowest densities, there was the highest silt clay content with over 97%, and the lowest sand content was less than 3%. Whereas station H and K, with the highest densities, had the lowest silt clay content with less than 94%, and the highest sand content between 6 and 7%. In order to determine whether the myofauna abundance is dependent on the sediment parameters, a Spearman's rank correlation was conducted and showed significant negative correlation for silt, clay, and total myofauna abundance, and a strong positive relationship between sand and chlorophyll A and total myofauna abundance. Hence, the remarkably low density at station C may be explained by the sediment parameters. We also wanted to address the question if metazoan myofauna communities differ in relation to the presence of nodules in the cores. For the nodule-bearing cores, we got 49 to 384 individuals per 10 square centimeter, and in the nodule-free cores, 37 to 352 individuals per 10 square centimeters. And the densities in nodule-bearing and nodule-free cores did not differ significantly. The myofaunal community composition showed weak significant differences in terms of nodule presence at major taxa level. Another analysis regarding the metazoan myo communities in relation to the abundance of nodules. Here the displacement volume was measured in the same way as, again, Annika did, and here we found no significant effects on the total myofauna abundance, and also not on individual taxa. So it's important, in order to understand the real community structure and its relation to environmental parameters, that the taxa have to be determined to a more precise level. Here we have only preliminary results so far, and my colleague, Dr. Ravel Singh, started to analyze the nematodes of the UK1 claim area stratum A to species level, and he reported that the most species are new to science and represented by 2 to 4 individuals per species. And he also observed 5 species present in the nodule crevices of the UK1 licensed area that are also reported from the German claim nodule crevices. So I will end by briefly giving you an outlook what we are going to do next. So of course we will do further identification of nematode taxa to the lowest possible taxonomic level. Then we are going to do some more molecular work. We have just started, and our aim is to integrate sequence data with a reference databases from other contractors. We also started to process and analyze the samples of the Abyss Line 2 cruise that has taken place this year. And then we would like to compare the data from both strata in relation to abiotic parameters to answer key questions about community structure and connectivity. I like to hand over to Aurélie now, and I would suggest if you have questions, you can ask me when Aurélie has finished her talk.
Well, good afternoon all. So I'm Aurélie Guano from the National Oceanography Centre and in the UK, in Southampton. And so I'm going to present the results dealing with the Atlantic foramifera, still in the framework of Lapis. So we attended the same cruises. So the first one in October 2013 and the other one earlier this year. And so for foraminifera faunal analysis, we have collected different kinds of samples. So first, we have collected sediment cores from megacore deployments from both strata. 1 core per deployment, which yields a total of 22 cores across both strata. And then we did live sorting of foramiferans and gromids during the cruises to collect specimens for morphological analysis, so that we preserved in formalin. So in total we have collected 335 individuals. And we also collected live specimens for single specimens genetic sequencing. And so for these samples we have collected a total of 892 individuals. For genetics, we also collected small sediment samples for next generation sequencing. And so for this, we have collected a total of 134 samples. So when we combine both cruises, we brought back to Southampton a total of about 1,850 samples, including about 1,000 samples for genetic sequencing and about 10,000 images of all the specimens that we have sorted for morphological and genetic sequencing. So, concerning the faunal analysis, so we collected sediment cores from megacore deployments, 1 core per station. So we try to remove first the nodules from the core, and we preserve separately the nodules in 10% formalin. And then we extruded the cores and sliced the core into 0.5 or 1 centimeter layers down to 5 centimeter or 10 centimeters. And we preserve each sediment layer separately in 10% formalin. Then when we went back to the lab in Southampton, we sieve each sediment layer through 300 and 150 micrometer mesh sieves. And then we stain the residues in a rose Bengal overnight. And then we sort all live, meaning rose Bengal stained foramiferans larger than 150 micrometer from complete residues. So all this work is the sorting is hand sorted. So this process is hand sorting of all the live specimens under a binocular. And we do this work on the top 1 centimeter layer of the cores. So, so we did this work on 5 cores from the first stratum A, and so this work is completed. It means that we have finished the sorting for the first cruise, and so far we have analyzed 2 cores from the second cruise. So for the results about the first cruise, in total we have sorted 4,465 complete live specimens, and the densities range from 73 to 165 individuals per 10 centimeters square. So it means an average density of 114 individuals per 10 centimeters square. In parallel, we have lots of fragments. So in total, we have sorted 2,000, a bit more than 2,000 fragmentary specimens, which is an average density of 55 fragmented specimens per 10 centimeters square. For the samples from the second cruise, we have quite similar trends in terms of densities, about 78 complete live specimens per 10 centimeters square, and about 41 fragmentary specimens per 10 centimeters square. So all these specimens, live and fragmentary, complete and fragmentary, live and dead, are identified to the species, to the genus and species level when possible, or classified into informal morphospecies. And so this is the kind of database that we end with, very complicated, lots of lines, lots of columns. And so to try to keep the things as simple as possible, we classify all our species and morphospecies into 22 formal or informal categories, and then into 2 major categories. So the first categories group what we call the conventional species, which are mostly multi-chambered and hard-shelled, and it's the kind of species that are very well known from fossil records and from modern environments, and that you can find lots of references in the literature and in the books. And the second group corresponds to the informal species, which are mostly single-chambered, and usually the conventional species, so the multi-chamber, the orange lines, they dominate the faunas. But in our sample, we have exactly the opposite trend. The conventional species represent almost 90% of the total assemblages, whereas so the single-chambered species represent more than 80% of the species. So in the conventional species, the most abundant group are the ormocinids, and in the unconventional species, the most abundant group is the tubular morphospecies. So over 5,000 specimens we have collected, 1,600 tubular specimens. So we try to keep track of everything. So we take photos of any single specimen and morphospecies, and we combine all these photos on a PowerPoint document to try to make a kind of catalog. And so this is not the last version, but one of the last versions of our catalog. So 201 slides, like these two, that combine all the photos of our 443 species or morphospecies that we have identified. And so among these 443 species or morphospecies, about 80% are undescribed and so new to science, often even sometimes at the genus level. And of these 80% of undescribed species, about 90% owns to the group of unfamiliar species. It means that there is really absolutely no reference and no record for that. So now some more results. So on this graph, we have plotted the number of new species identified in each sample versus the number of total specimens. And so, as we expected, the species list grew with the addition of new samples. And so, well, on average, we discover one new species every 8 specimens. And if we plot now the rarefaction curves, we see that we are far from reaching any asymptote or level note, which means that definitely there are still more and many more new species to discover. One point that is not illustrated on this slide is that many of our species are represented by very few specimens. On average, about 10 to 30 species are represented by more than 5 specimens. And often also some species are only observed at only one station. So now about the molecular analysis. So we have 2 kinds of samples. Samples for next generation sequencing. So we collected small sediment samples from megacore sediment cores, 3 replicates per core and 2 cores per station. And so we preserve these small sediment samples in live world. And for single specimen genetic sequencing, we also collected mostly the specimens from megacore samples, and we preserve the specimens in RNA data or guanidine. I think one important point to precise for these samplings is that we perform all these samplings at low cold temperatures. So it means either sampling in a cold room at about 2 degrees and sorting of live specimen on ice. And this is to preserve the DNA of the forams. So among the almost 900 specimens that we have sorted for single specimen genetic sequencing, we've tried to amplify 306 specimens belonging to different groups: Comocasans, Saccharines, Allogomies, Nodelum-like, Calcareous, and Hormocinids. And the success rate is quite variable. Nodelum-like, none of the specimens that we tried to amplify worked, unfortunately. But Hormocinids, 100% worked, but we only have 3 specimens. The interesting thing is that we have quite a high success rate for the Comocasans, about 37%. So far, there is absolutely no sequencing for this group. But, well, the amplification worked, but we don't know if the sequencing will work. And hopefully, we'll have an answer early next year. So this work is done at the University of Geneva by Maria Holzmann. And so for next generation sequencing, we made the amplification of samples from 7 stations from Stratum B, so meaning from the second cruise. So 2 cores and 3 replicates per core. So in total, 42 samples, and so these 42 samples are waiting for the extraction of the PCR of framiferal and eukaryotic DNA and RNA. And so this work on the NGS samples is done by Frank Lechzerowicz, also from the University of Geneva. So yeah, this is one of our collaboration with the University of Geneva. So it involves Professor Jan Pawlowski and Dr. Frank Lechzerowicz and Maria Holzmann. And we also have another collaboration with Sheshok Institute, Moscow in Russia. And this is with Dr. Olga Kamenskaya, and this collaboration is related to the identification of Comocasans. So I will just finish by some problems that we have with the analysis of the forams. The biggest problem is obviously the taxonomical identification. So, well, first, the sorting of the samples is really time-consuming because the volume of the sediment is very big and the densities are also very high. We also have many species which are prone to fragmentation, which makes their identification very difficult. And so we have 80% or so of species which are undescribed. So, and we need to compare any single specimen with previously identified specimen in order to stay consistent with our taxonomy. And so this is really time-consuming and challenging. So in the future, we want to try new approaches to try to find a way to analyze the samples more quicker and maybe to reduce also the densities of the forams. So either focusing on a finer fraction down to 63 micrometer, but on a smaller samples, so by splitting the samples. And so hopefully, well, we hope that we will get more conventional species that could make the taxonomy easier and so the analysis of the samples faster. Thank you.
When you sort you sit your sample on a 150 and a 300 micrometer?
Yes.
Does that mean that you look at the fraction in between, so smaller than 300?
Yeah,
larger than
yeah.
100?
We have a look first on the 300 microns residue, and then on the 150 to 300 residue.
Okay, so the 300 fraction is included.
Yes, we include. Yeah.
Okay. And I didn't fully understand why going to a lower sieve would facilitate or make the work easier, because it would mean you have more sediment.
Yeah, no.
More difficult to store.
So we over the 5 samples from the first cruise, we have sorted 3 complete samples, and then the 2 last samples we tried to split. So I think that we analyzed about 3/8 of the sample, and so yes,
smaller sample size.
Smaller samples, yeah, and so less individuals, and so,
okay. But on a 63 micrometer sieve, I can imagine you have all the sediment, so it really goes very slowly to.
Yeah, and so if we go down to 63 micrometer, we will probably work on only 1 split. So we will get less specimens larger than 150 micrometer, but we will get the specimen from the final fraction. So maybe we will get in total the same number of specimens to sort, but we think that maybe we will have more conventional species in the 63 micrometer fraction. Because in this fraction we used to find, for example, the opportunistic species, like some calcareous species, that we don't get in the coarse fraction.
I have a question for Inga. And your talk, you had a slide of comparing abundances. I wonder if you could fire that up. Probably that one. That we just ask. Next one. That's it. And maybe Gordon's going to be hot on this. I don't know. We were sort of in the macrofaunal meeting, and it's about statistical power, and as well raised this about increasing the number of samples to give better statistical power. Because the problem is that when one has this conclusion that there's no statistical difference, he can take that away and say, oh, there's no statistical difference. But it's more to do with that we just haven't taken enough samples than it is that it's a real difference.
Yeah.
So I don't know if this workshop wants to consider as well what statistical power means for taking samples from myofauna, because you may come to the conclusion that myofauna can't be used ready for monitoring because you just have to take so many samples that, well, why bother, I suppose is the question. So that's just something I'd like to raise.
Yeah, but I totally agree.
From your.
And it also would be interesting at a higher taxa level. So then this is only at major taxa levels. Yeah. And maybe then we can see significant differences.
Okay. Thank you very much.